Hello and welcome to the second webinar on the use of integrated photonics in healthcare, this time about biosensing. I'm Karel de Vries. I'm responsible for strategy and startup development in PhotonDelta, and I'm really hoping you will have a very interesting presentation here. We do this together with TNO and the University of Eindhoven. And this is a series of webinars. We plan the next one after the holiday period. I will introduce quickly to you what PhotonDelta is and does, why integrated photonics can revolutionize healthcare, what is already visible and available, and why this is the right place to be if you want to be up to date on what's happening in integrated photonics for healthcare. Then we have 4 pitches of 4 startup companies: Surfix, Delta Life Science, each active in different areas of what is called biochips; Nostics, which is looking at the use of Raman spectroscopy in healthcare; and Amazec, which is an application of fiber Bragg gratings in healthcare. Last webinar you already saw what the technology can do in vital statistics. This time you will see an application in biosensing. We will end with a question session, so the request is to put your questions in the chat. Uzunbajakava from TNO, our expert on technology, photonics technology and healthcare, will gather all the questions and will lead the panel. First, what's PhotonDelta and what is it doing? Well, first of all, PhotonDelta is a cooperating ecosystem of companies that are active in integrated photonics and R&D organizations that drive the innovation there. But PhotonDelta is also an organization, and I'm part of that organization. Our role and target is to do economic and ecosystem development of integrated photonics in the Netherlands and on a global scale. We are most well known for the fact that we are responsible for the managing of the 1.1 billion euro growth fund program that we got on behalf of the Dutch government, and where we run extensive programs with a lot of our partners. But we are also active in international and in EU relationships. We help with market and talent development, and last but not least, we do startup support, and we have a seed investment fund to invest in very early phase startups in integrated photonics, and a number of them are in healthcare. We also want to have what we call complete supply chain in our ecosystem. So in other words, if you happen to be somebody that wants a healthcare solution, you want to have somebody to design your photonic chip, you want to have somebody that can actually produce the wafers, get them packaged, and develop applications. There are quite a few years ago we started with only a few companies. Now we are more than 70 plus several dozen of companies that support the development of system solutions, and a large number of institutes, of which TNO is the place which is hosting this session, but also Amazec is a very strong partner in this as well. So you are essentially able to get a complete end-to-end solution in our complete ecosystem. Now you can say, fine that I can get that, but what is so relevant about integrated photonics? Why is it potentially revolutionizing various areas and potentially also healthcare? Well, you might think about the photonics part that this is an optical activity, but essentially we are in the semiconductor industry. Integrated photonics is using semiconductor technologies and semiconductor processes, semiconductor factories, in order to get chips made, which the big difference is that we use photons instead of electrons to get things done. It means that optical functions get integrated in a chip-like matter. That has several effects, and one of them is that it can become quite small. By integration it is reliable. You can reduce power consumption a lot, but a very important effect is that it can also become scalable and suitable for producing at high volumes and thereby lowering cost. So in healthcare, this translates into enabling sensors, mostly for instance, wearables and point-of-care solutions where volumes and small size and low power are important. There are also a number of high-value applications where the chip really has an added value, but this is really similar to what's driving the semiconductor industry is driving the integrated photonics industry for healthcare. We have a quite complete set of technology platforms for health, and that's sensing platforms. A few years ago, 5 years ago, we started with only one, fiber brake gratings, and Pim, who will be talking to you later, was one of the founding fathers of that technology. In the meantime, we have more than 10, and there are still technologies added to the list. So, in the meantime, I get the point that if it's worthwhile doing, you can put it in a chip. And that some you can already buy, like fiber brake grating solutions, like the near-infrared spectrometers. There are others you will see. So others are in the area of getting ready for industrialization. Biochips is certainly an area in that, very close in that, but some others are as well, and you might have heard some of that in the previous webinar. So altogether, the scope of solutions is widening quite, ranging from spectrometers to gas detection systems in different areas, to systems where you can measure blood flow or measure optometry, the eye care or dermatology. The other reason why it's interesting to be around here is that this is a hotspot for integrated photonics. So this region has a large number of companies active in integrated photonics, but also a lot of people who have knowledge about healthcare, which make this an interesting place to start talking about solutions. And as I already showed you, we have a complete supply chain to support development and production. We also can give extensive support. This region, the so-called Brainport region, has many companies, but also a lot of institutes like TNO, TOE, Amazec, that can support development, but also organizations that can help you setting up activities and companies. We have the photonics ecosystem, and of course PhotonDelta is the organization that is really helping to support you in getting around in there. And the last but not minor point is that we have startup support, also working together with several companies, venture builders in this region and others. And we have our own very early phase seed fund, but also a lot of contacts with venture capital companies that can help you to set up activities or companies. And with this, I've ended my introduction. So the next presentation will be from Surfix. I would like to invite Jos Liebenberg, CEO of Surfix, to the stage, and he will give an introduction into what biosensing in biochips can mean for your industry. Jos, take it away.
Yes. Thank you very much. Good afternoon, everyone. My name is Jos Liebenberg. I'm the CEO of Surfix. Since August 2023, we are on a quest to really redefine point-of-care testing with a photonic diagnostic platform, and I will show you that we don't only claim it, but that we also have proven it. So our mission is to advance global health by enabling low abundance biomarker testing at the point of care, redefine diagnostic, and unlock a large market share for disease detection and management for point of care POCT. So our unique set of USPs unlocks this low abundance biomarker detection at POCT, a currently unserviceable, large and significantly growing lab-based market segment, because we have proven that our lab level sensitivity and precision in detecting proteins is at sub-picogram per ml level for proteins. We can also do the multiplex with the current design of the chip up to 5 results are obtainable in 10 to 15 minutes compared to lab systems. These centralized lab systems up to 300 times faster if you include shipment. And it's multimodal, very important. We can not only service immunoassays but also molecular assays on one instrument. And I'll give you details in a bit. Our costs of goods are kept very low because we have developed a dicing technology at wafer scale. So what makes us so unique? A picture says more than 1,000 words. Here you see the 5 top players that also have sensitive systems in point of care. However, we stand out and win on the sensitivity part, on the broad dynamic range part, which is very important if you want a multiplex, that you not only be able to pick up those low abundance markers, but also markers at, let's say, microgram, nanogram levels. And that combined in the capacity to multiplex up to 5 is truly unique, because others do not have any multiplexing capacity, as you can see on the right hand side of this picture, or up to 2 or 3 only. So the combination of what we do is quite unique, and it has been successfully validated by top-tier diagnostic players in blind studies. And yes, this multimodality offers enormous market potential, but as a small company, of course, focus is key, and we will focus on those low abundance immunoassays at first, because there you see a perfect market fit. Our total addressable market, the global, let's say, point of care diagnostic markets, is moving to 79 billion in 2030 with an average annual growth rate of about 12%. And other areas to really unlock the broad spectrum here in this market are gradually added over time, like the molecular markers and molecular assays. Okay, let's zoom in a bit on technology and why we can do where others fail at the point of care. Well, it's by distinctively combining 6 state-of-the-art technologies. And the starting point is, of course, the chip. And our chip has 20 years of development in it. It's the best-in-class chip. It's got, through its design of self-referencing and extremely long detection areas or waveguides, respectively a elimination of the matrix effects and a very high sensitivity in itself. But we have a history of being a CRO for nanocoatings, and those 10 years of knowledge gained are still very beneficial to everything we do in product building, because we have shown that by applying a nanocoating selectively on the waveguide versus all the areas of the chip, we can, at low analyte concentration, boost the detection levels up to 10 to 100-fold. And then the bio team. We have the expertise in-house to quickly do the feasibility studies. Very fast, we can find out whether your particular biomarkers can work on our chip technology. Very important, of course. Then the cartridge technology. We've gained a lot of experience over the years, also through the nanocoating years, in partnerships with key companies in that area and academia. And this has resulted in very smart application of microfluidics. And as said, we can warrant low cost of goods because the dicing technology that we've developed allows us to do the coating first, then adding the biotechnology layer, and then only then do the dicing of the chip. And that again is very scalable and allows us to move to very low cost of goods. Then the last key element is the point of care instrument. We have a great collaboration with a very experienced instrument builder, and we tick all the boxes for point of care. Okay, let's zoom in a bit then on the development plan, manufacturing plan, because currently it's 2025, we have our functional model of the generation 1 instrument available, and stepwise we move it to a very small, fast, low-cost point-of-care solution, as you can see in this graph. The cartridge production follows the same path, and we will have an in-house low-volume production line available by mid-2027. So all the key elements that we need to move the solution to a final stage have already been sorted and are in scope. So let's look at the game plan here. Partnering with top-tier diagnostic players and KOLs in hot spaces is the aim of this game. We do paid technology evaluations followed either by grant projects or assay co-development. And that can be with top-tier players or with KOLs as said. So over the years we do a lot of value creation through these strategic partnerships. And it's in action. It's not the aim, it's live. This is an overview of where we stand at this moment in time. As you can see, we have ongoing or already concluded projects in 3 different important market segments: infectious diseases, oncology, neurology, and also at different stages of disease, from screening to monitoring. I can of course not disclose all the names, but you see 1 or 2 projects are in collaboration with the Netherlands Cancer Institute. So, in summary, Surfix can own a rapidly growing unserviceable market segment in POCT of those low abundance or low concentrated proteins. We have early top-tier clients, some of them already in large follow-up projects. Our technology has been validated by them, 2 top-tier diagnostic companies, and with that we can claim that the technology has been largely de-risked. Co-development as a business model is illustrated and in execution as I showed you, and we are in a great position with that to make the immunoassay market at POCT level explode. With that, I would like to thank you for your attention, and at the end of this session, after 4 pitches, I'm happy to answer all your questions. Thank you very much.
Thank you, Jos. Please have a seat, and I really like the type of developments you showed here. Next one on this is Bart de Boer. Bart is also working on a different way in the area of biochips, and will tell you all about the interesting developments that he is going to bring to market.
Hello, my name is Bart de Boer. I'm the CTO of Delta Life Science. Let me start with a quick introduction of Delta Life Science. We develop biosensors for life science research. We are a spin-off of TNO, the Dutch National Applied Research Organization. We were founded in 2018, and we have offices in Rotterdam and Enschede. And we have a team of about 20 people working in engineering, developing our products, in a wet laboratory to validate our technology, and also to perform feasibility studies with our customers and in sales. So let me start with the problem we address. In the development of new drugs and diagnostic tests, there are literally thousands of experiments that need to be conducted to detect and analyze the binding between 2 biomolecules, between an antibody and an antigen, between a drug and its target. And for that you need biosensors. And to do this efficiently, there's a need for biosensors with ever higher sensitivity to be able to detect even the smallest molecules in the lowest concentrations, and a higher throughput to analyze as many samples as possible in the shortest amount of time. Now, the conventional technology used in biosensing has been around for 25 years, and it has played an enormously important role in life science research. But at the same time, you see that this technology has now reached the limits of its capabilities. So today's answers are not yet answered with today's questions are not answered with the current technology, and there's a need for new technology. At the same time, we see that big advancements have been made in the field of integrated photonics, or optical chips. Thanks to huge investments that have been made in the last decades, we now have access to a very mature technology that can be used to drive products, and that we have used to develop a biosensing platform that in performance outperforms conventional technology. So these two factors, the timely maturation of this new technology, as well as the urgent need for biosensors with an ever higher sensitivity that comes from the life science research industry, have inspired us and also enabled us to develop this new biosensing platform. This picture shows the surface of one of our optical chips. Light is transported in waveguides and interacts with any biomolecules that bind to the chip surface. And that interaction changes, in fact, the speed of light, which light travels in the waveguides, and this can be accurately measured with our instrument. Now, this technology has a number of unique features. It's ultra-sensitive, which means that our customers can detect the smallest molecules in the lowest concentrations. But it also means that we use lower amounts of precious samples. And it reduces the time it takes to perform an analysis, reducing the time it takes to perform a study. Now, we can put many of these sensors on the same chip surface, which means that our customers can detect many different biomolecules in the same sample. And that, again, increases throughput and also reduces the time it takes to perform a study. And finally, this technology is extremely affordable compared to conventional technology, which means that biophysics labs can reduce their capital and operational expenditure greatly. Now, with this technology, we have launched our first product called InQuick, which offers high performance detection of 16 different biomarkers at the same time in the same sample. And it is able to analyze up to 10 samples sequentially in a single unattended run. And we're quite close to finalizing our second product, which will allow users to analyze up to 768 samples sequentially in a single unattended run. So I would like to show two examples of how such an instrument is used in practice. And the first one is analyzing binding kinetics. You can imagine that if you want to select an antibody for use in diagnostic tests, you want to know how fast the antibody is able to bind to a biomarker and how long it stays connected to that particular biomarker. And this is exactly what we study when we perform analysis of binding kinetics. So what is shown here is a typical sensor response as function of time during a binding kinetics experiment. We immobilize the antibody on the chip surface and then start by measuring a baseline, by measuring a buffer solution. Then we switch to a sample containing the biomarker. We see an increase in the signal, and we can follow that in real time as a result of the binding of more and more biomarkers to the antibodies on the chip surface. Now, at a certain moment, the signal plateaus. This is equilibrium, and then we switch back to buffer solution again. And then we can measure how fast the antibody releases the biomarker again. And this is shown in a reduction of the sensor signal. And then finally, from the shape of these curves, we can determine the binding kinetic properties. And from that, we can determine whether or not the antibody is suitable for use in our diagnostic test. Now, you can imagine that this is also important in drug development. Also there, it's important to characterize the interaction between the drug and its target. So both in drug development and diagnostics development, these types of measurements are performed. But they also find many applications in basic research. The second example is biomarker quantitation. In diagnostics, but also in life science research, very often we need to determine the concentration of a biomarker in a sample. Again, here we immobilize an antibody on the chip. And in the left graph, we see the sensor response as function of time for multiple experiments with different samples with different concentration of the biomarker present. And we can detect the concentration of the biomarkers very fast in minutes time frame. In fact, we don't even need to wait until the signal plateaus because even the slope of the signal increase after the introduction of the sample is concentration dependent. So based on this slope, we can immediately determine the concentration of the biomarker in the sample. Now, once we've performed several experiments with known biomarker concentrations, we can make a dose-response curve, which relates the biosensor signal to concentration of the biomarker. And once we have that relation, we can start measuring samples with unknown concentration of the biomarker. And we can then, using the dose-response curve, determine the concentration of the biomarker in the sample from the sensor signal. Again, this is shown here for one biomarker, but remember that we have 16 sensors on our chip, which means that we can detect 16 different analytes in the same sample at the same time. So to conclude, integrated photonics has enabled us to develop a biosensing platform that outperforms conventional technology in terms of biosensing. And it also allowed us to develop a system that allows our customers to reduce the cost and time it needs to perform their studies in the development of drugs and diagnostic tests. If you'd like to know more about our technology and like to follow our progress, please visit our website and follow us on LinkedIn. And please contact us whenever you would like to. Thank you for your attention.
Thank you, Bart. And again, you made the point that you can get more information later on. We intend to make both the webinar publicly available on YouTube, as well as that the slides will become available through our PhotonDelta website shortly. So we will give you every possibility to find the material back later. Please put your further questions in the chat if you have them, because that we will be able to discuss that later on. And that means that I now can introduce my next speaker, Alexander van Reenen, on a totally different application of photonics in healthcare and Raman spectroscopy. Alexander, please take the stage and guide us through this.
Thank you for the introduction. So I would like in the coming 10 minutes to present to you our company Nostics. And we at Nostics, we have the vision to democratize diagnostics. So we want to make diagnostics available to every corner of the world. And therefore it needs to be affordable. But you cannot compromise on quality. And it needs to be timely. So the diagnostics need to be also rapid and reliable. So one of the problems that we address is the problem that in the field of infectious disease, during a typical doctor-patient interaction, there's only 10 minutes time for a doctor to take a decision. But for infectious diseases such as urinary tract infection, it takes typically 2 or 3 days to get a test result from the lab. So that means that because treating an infection is urgent, doctors decide on prescribing antibiotics before they even have the test results. And this leads to ineffective patient treatment because 20% of patients that need antibiotics receive the wrong treatment. And this worsens their disease state and causes higher healthcare costs. On the other hand, generally you also see there's an overuse of antibiotics just to be on the safe side. But that means that in reality about 50% of antibiotics are prescribed unnecessarily. And that will cause resistance and will make treating infections even more ineffective and eventually impossible. So what is the solution that we work on within Nostics? Well, we work on a solution where we want to confirm and identify bacteria in less than 10 minutes. And for that, the test needs to be easy to use, to be able to use it in all sorts of settings. So our system consists of two simple components. On the one hand, a disposable cartridge to which the sample is applied by the user. And subsequently, the cartridge is inserted in the instrument, where it's automatically processed and the result is analyzed by making use of AI. And this results in actionable results for the doctors, how to treat the patient, if any treatment is necessary at all. So delving a little bit deeper in the technology that we use for this, we make use of a relatively old technology, Raman spectroscopy, and this is based on Raman scattering. And generally, if you try to detect a particular molecule and you have an incident laser light at a certain wavelength, most of the photons scatter at the same wavelength. But 1 in a million photons will scatter at a different wavelength because of the small vibrations in the molecule that cause a slight wavelength shift. And depending on the molecular structure, you see different shifts. And it results in a so-called fingerprint, a Raman fingerprint of that molecule. But this technology alone is not sufficient to enable detection at very low concentrations in a short amount of time. So typically you need some sort of multiplication like cell culturing. However, at Nostics, we make use of nanomaterials like golden nanoparticles, which we aggregate, that create hotspots that cause the electromagnetic energy to concentrate into those hotspots where the molecules are. And that amplifies the signal dramatically. And collecting these amplified fingerprints, we make use of AI tools to extract which bacteria is detected into these hotspots. So comparing ourselves to other systems or tests that are available, well, first of all, to win in this space, you need to combine performance with time to result. So you need to be able to accurately detect and identify bacteria. And you need to do so in less than 10 minutes during the patient interaction that the doctor has. But also to be able to use it in decentralized settings, it needs to be operable by untrained operators. So everyone should be able to run the test. And lastly, it needs to be affordable. The golden reference currently used is the dipstick test, which has all those last 3 components, but really lacks the performance and is not able to identify which bacteria is present. And if you look at systems that are able to achieve that performance, then you see that the time to result is way more than 10 minutes. It's generally maybe 1 hour or much longer. And those systems require laboratory skills to be operated. And eventually, it involves expensive equipment, which does not allow the system to be affordable for most settings. But we built this system as a platform, and with the platform we can test different applications. And the first application to which we will focus is the urinary tract infection. And here we will launch a test system that enables to detect a panel of 9 different bacteria that covers all the UTI cases in primary care. However, you can easily expand this. We can easily expand this platform to different diseases like bloodstream infections, fungal infections, sexually transmitted diseases. And also we're currently working on feasibility for AST, so antimicrobial susceptibility testing. And that is also of interest because if you know which bacteria is present in the sample, you might not know to which antibiotic it is resistant to. And getting that information as well enables the doctor's decisions to be even more effective. But we achieve an excellent test performance by making use of AI, and we use large spectral models. And this in this approach, we make use of a foundation model where we compile a huge data set of different spectra on which we train the model to identify the different main changing features in the spectra. And we focus for a specific application on task-specific data. So for instance, the UTI bacteria. And by making use of the foundation model, we only need a limited set of data to train the model. And using that model, you can see on the right, we are able to achieve an accuracy for the identification of bacteria of 91%. And if you weigh it on the prevalence, it's even more, it's 97%, which means that we're able to detect the more common bacteria even with a higher accuracy. Our detection capability, we've also shown it's almost 90% in clinical urine, and we're currently working to improve that by optimizing our cartridge design. On the right, you see an image of the instrument prototype that we currently have. And here the main focus is to integrate a low-cost spectrometer in a benchtop system that is portable. Some details on the application, the business opportunity that we have. Well, the infectious disease market is quite big, as most of you will know, but the UTI market is a relatively large part of that. So the incidence rate is about 400 million per year, and we intend to first launch in advanced countries because of the higher accessibility and focus on the high revenue accounts. And there already we have a business of over 1 billion. Also there we can access smaller accounts, and once we have launched, we will also focus on expanding into emerging markets and then extending the platform to expand to other infections. Currently, we're seeking funding for our Series A in order to complete the development of our UTI application up until clinical validation, to further optimize the large spectral model on our system, and to transition from cartridge and instrument prototypes to scalable manufacturing. And lastly, startups like this are not made of a single person, but are made of a broad team. And our team has a broad set of experience, both on the entrepreneurial side and also on the deep domain side. And together, we work to make this happen. And with that, I would like to thank you for your attention and also for the opportunity to present here.
Thank you, Alexander. This was again a very interesting application of what is possible with photonics in the area of healthcare, and hopefully indeed in bringing forward new possibilities in affordable care. The next one, so thanks again, and the next one is that we'll be seeing is Pim Kat. If you have been following the first webinar, you might have already seen a few things on fiber brag grating solutions. Pim Kat was effectively the founder of what is now Photon First, and he couldn't resist going on and finding new possibilities for this exciting technology, and he started Amazec, and Amazec is using this technology for very new solutions in healthcare. So please, Pim, take it away.
Yeah, thank you for the opportunity to tell what we're doing in Amazec Photonics. I'm Pim Kat, CEO of the company and one of the founders of the company about 4 years ago. We use fiber brag grating sensing for high resolution temperature sensing. And why do we use this technology? It's mostly about the resolution that we can get with a chip that you cannot reach with standard optical systems. So the number of sensors can be high, resolution can be extreme, and the speed is mostly depending on the electronics that we use. But we think we can reduce the prices of complex systems to an affordable level. And what we do in Amazec Photonics is a procedure to detect heart failure by thermal dilution. That's technology that's already used for decades. Basically, you inject cold fluid in the bloodstream, 10 cc of saline, 4 degrees C standard, and we measure the throughput through the heart by measuring with a thermometer at the outcome of the heart. So typical curves show you only one pass of the cold fluid through the heart, and if it's very fast, it's a very good functioning heart. If it's very slow, there's probably something wrong in that. But what we try to do in the new company is even measure the recirculation of this cold fluid after one full pass through the body. Of course, it's not only one path that the bloodstream will follow, it's a combination of parallel flows. So you see the red curve here is the first pass through the heart. That's the thermal dilution curve that every system can measure. And what we expect is to see recirculation of the cold fluid through the heart, through the liver and the kidney, and through other organs after some time. For that, we need a resolution at the level of 0.1 millikelvin, and that's why we use integrated photonics to reach this extreme temperature resolution. It equals 1 femtometer wavelength resolution at 1550 nanometer wavelength. So how this looks in the body, you can see the injection going into the blood flow. It goes through the heart, through the lungs, through the heart again, then divides into different streams, passes the first sensor, the first pass through the sensor, goes through the whole body, again through the heart, through the lungs, through the heart again, and then after some time we see the second passes of all the blood streams following after that. So that was the basic idea, and we were pretty sure that we could see this pass of the cold fluid second time through the heart. And this is one of the first experiments that we did non-invasive with a skin sensor on the pulse, hoping to see this first pass of the blood, the cold fluid, and the second pass, the recirculation in the bloodstream. But what we saw mainly was the reaction, a skin reaction by injecting the cold fluid in the bloodstream, and that's called chicken skin, and that's basically what we see on the sensor. So there's a lot of work to be done to get the right signals from the recirculation from what we see. So we started doing something else, and this is the second measurement that we did. It's in the esophagus, it's in your throat. This is what you see in the picture is a sensor that is used also to look into the heart function with ultrasonic. So you can see a small sensor on the top of the ultrasonic probe. That's our thermal FBG sensor, and we try to pick up the cold fluid signals directly on the side of the heart, so in the esophagus, in your throat, and measuring through the heart wall. But unfortunately, we didn't pick up any signal on that probe, so a second disappointment. And we went back to a third trial. That's the invasive measurement by injecting in the vena carotis and measuring in the vena renalis. This is a pig on the table. It's animal tests that we do. And what we found here was a very strong and very clear first pass of the cold fluid through the heart. And the recirculation was detected in the vena renalis. That is after your that's the division between your aorta and the stream through your head, and it was even detected by measuring after the kidneys. So, and that means that at least we can see recirculations of this cold fluid through the heart, all with the focus of measuring this recirculation and being able to calculate the total circular bloodstream in the human body. And that's something that a lot of people have already been looking for years. There's no feasible way to do this at the moment. And we can detect the total circular blood volume within some minutes. And that means when people come into the hospital with blood loss or during operation, they lose a lot of blood. There's no way to tell exactly how much blood is still in the human body. And after having this process in place, we can at least tell within some minutes how much blood is still going around in the body. And that will save a lot of lives. So yeah, that's up to now with the sensor for Amazec Photonics and doing the heart failure detection. In the meantime, when we had this system ready, the system was detected by or found on the internet probably by a company in high resolution or wanting to do high resolution sensing in semicon processes. And we are now working on sensor systems to detect temperature and strain on an extreme resolution. We're talking about sub-nano strain and sub-millikelvin resolution on multi-points on a full wafer. So Amazec keeps focus on the business for cardiac failure and circular blood volume. And we just founded a new company using this same technology, same system in Ionics company. Focusing on measuring first in semicon processes on full wafer scale by using fiber break rating technology, ring resonators, and DBRs, distributed break sensors. So that brings me to my last slide already. I want to thank all the people that invested and helped us getting to this point. And yeah, that's what I wanted to say. Thank you for your attention.
Thank you, Pim. I think one of the interesting takeaways is how extreme accuracies can be achieved, and I hope this will inspire others on the webinar, perhaps, that to see whether they have interesting applications for millikelvin measurements or sub-nanometer accuracies somewhere else in healthcare. We have shown that's possible. Yep. So sometimes you find more things than you started out looking for. So thanks again. Well, during the sessions, questions have been coming in. So we will now start the questions panel, and Natalia will lead the panel. So I hope you have gotten interesting questions. And,
yes, yes, absolutely. Questions were flooding into the chat, and we collected them all. So now we can drill a little bit more on this wonderful topic of integrated photonics for biosensors. So we have wonderful panel members. Is Pim Kat from Amazec Photonics. We have Alexander van Reenen from Nostics. We have Bart de Boer from Delta Life Science. And we have Luc Lunenberg from Surfix. So let's start with a few questions for Surfix, Luc. A few people are asking in the chat, how can you elaborate a little bit more on surface functionalization that you are performing? Do you develop chemistry in-house? And in general, what kind of challenges do you see when integrating functionalized chips in a cartridge?
Mm-hmm. All right. Yeah, Luc Lunenberg makes me the CTO and the CEO at the same time. Unfortunately, my name is Jos, so I don't have the technical background to maybe answer this in all details. Still, for the surface part, I would say the key element in what we do is in the fact that we apply a nano coating selectively. On the waveguide, where we can measure, we have an attractive layer. And in all the other areas on the chip, we have a sort of anti-fouling. Don't know if you're a sailor, but on my boat, I have an anti-fouling underwater jet that pushes away everything that wants to grow there. So all analytes will be pushed away until it reaches the waveguide, and there we attract it. And that again is one of the secret sources of our high sensitivity.
Yeah, so
And the integration, I mean, the way we currently do it is very important, of course, that it's well positioned, and we have a so-called pick-and-place machine in place for this in our in-house production facility.
wonderful. So I hope the listeners are happy with these answers, and they keep asking questions. What type of sample can be analyzed using Surfix technology? Can it be whole blood, serum, something else?
Yeah. So what we currently focus on, basically, let's make one step back. As said, one of the effects of the design of our chip, we are pretty much matrix independent. The self-reference part makes this. So we can basically analyze any bodily fluid. However, focus is key for a small company. We concentrate on indeed blood and plasma. We will do a sample prep on the cartridge with a filter and run the plasma as such in the microfluidic channels.
Wonderful. Do you need funding, maybe? How is the situation?
Don't we all need funding at a certain moment in time? Yes, we also need funding. We're well on our way to close a larger, call it Series A, of about 50 million. We have a term sheet drafted. We have a LOI of a lead investor. But if there's anyone in the audience that know investors that would like to join us in this consortium that we're building, I'm happy to receive the names or your personal message.
Wonderful. Thank you so much. Jos, I think you are answering questions better than Luc. And there is a transition question actually to Delta Life Science and to Surfix. And the question is the following: can your technology deal with low and high abundant biomarkers in the same sample running in multiplex mode? Thank you for interesting pitch. So yeah, how can we deal?
I can, but it was your question, wasn't it?
Up to you. You both.
We can both answer.
You can both answer.
You go first.
Yeah.
So I would have to say it depends, of course. You would have to test it to see. In principle, it's possible to detect low abundant and high abundant analytes in the same sample. Of course, it all depends on your assay development and how well you're able to develop the assay for that particular application. But in principle, it's possible. Yeah.
Yeah. Jos, would you like?
Yeah,
to?
same for us. One of our first showcases was on sepsis, and there's 3 very good biomarkers that are on very different levels, from picograms to nanograms and micrograms, and we've indeed shown that we can run them in one test and be able to multiplex low and high abundance markers at the same time. Yes.
Wonderful.
But it's completely true what Bart says.
Yeah.
It all depends a bit on.
Yeah. And Bart, a little bit technical question, another technical question. So how about your sensor? Is it disposable? Is it reusable?
Yeah.
Can you elaborate on that a little bit?
Yeah, that's a good question. So the sensors are consumables, but you can reuse them many times. So the instrument uses a regeneration fluid to regenerate the sensor surface, which means that after each experiment, a regeneration fluid is flown across the surface, and that releases the analytes from the chip surface, which makes it again suitable for the next experiment. And that is of course needed, because if you want to measure 768 samples sequentially in a single unattended run, you need, of course, to be able to regenerate the chip surface between the experiment, and that is done by the instrument itself.
Okay, so. Wonderful. So a very smart solution. Thank you very much, Bart. Questions to Nostics, to Alexander. Can you elaborate if you need to functionalize the plasmonic surface for your target molecules?
Yeah, I can elaborate on that. So we don't need to functionalize the plasmonic surface. So what we do is we basically collect the bacteria at a filtering surface, and we're not detecting the bacteria directly, but we're sensing the components that are excreted by the bacteria upon stressing them. And by drying the bacteria in, for instance, on the filter, the components that they excrete will get dried into the hotspots of the source particles. And to this effect, you concentrate them in those corners, and you can then sense them. And in particular, also the amplification is large. It's really over 10,000 times. So the signal that you get by using these plasmonic surfaces is really substantial.
That's wonderful. I think that's really the power of your technology, Surface Enhanced Raman, and the way how you challenge the bacteria to produce specific components. Do you have, for example, ideas about what will be the cost per one test unit of your technology?
Yeah, so first of all, the sales price is going to be important, and that really depends on the reimbursement for these specific tests. For the UTI business case, it's useful that there are already tests out there, so that more or less set the bar where you need to aim for. And for a viable business, we need to stay well below it to also make it attractive to invest in this. So generally, I cannot give exact numbers, but you really want to go much down below the, well, let's say at least below the $5, but even further at scale, because it depends, of course, on the scale at which you start producing it.
Yeah, wonderful. So it looks like you are really on a good way towards addressing global challenges with affordable solution that can be accommodated by the target group. Pim, Amazec Photonics. So the question I have, how do you stay so proliferative in generating based on the technology?
Well, maybe you saw the logo of the last company, and as you know, my surname is Cat. So on the logo, you see the look on the face of my wife, and I told her I would start a new company again. But basically, what you see is so many opportunities, and it's so, it's a pity to just let them go away. But what I'm doing at the moment is finding young, enthusiastic people to take it out of my hands, and I will just be an investor and advisor from, I hope, 1 year from now. But there are so many things that are so promising and so new and so interesting in integrated photonics. I couldn't stop.
This is wonderful. I think this is wonderful summary of our discussion, Karel. So how can integrated photonics revolutionize healthcare? What's your pitch on this?
I think could be no nicer than the last two answers to the questions. One is there are possibilities to go at scale and go to price points that were hitherto totally unavailable, levels that were also unavailable. So, and then Pim tells me there are so many opportunities that you can start up almost every day, and that's what we would like to support. So we can revolutionize healthcare by simply go about and do it.
Wonderful, and thank you very much. Very experienced, very engaging speakers, wonderful pitches, and thank you very much, audience, for being with us and asking all the questions. Till next time.
Thank you.